Thank you, Bob. So, in my turn, I would like to talk to you about identification of complex oxidized lipids from LCMS dataset. So there were a lot of very amazing talks before me about eicosanoids, so oxidized fatty acid in principle, but in my lab we are very much interested in also oxidized products or modified products from the complex lipids. So we believe that lipid modification, when present in nature, are very important mediator and regulatory molecules increasing capacity of biological systems. So those complex lipids can get modified on different positions, including their head groups and fatty acid chains, maybe sometimes even a backbone, via introduction of small chemical groups, enzymatically or non-enzymatically. Just a few examples here. For instance, a PA-PE lipid can get oxidized via lipoxygenase catalyzed reaction or free radical driven hydroxylation with the formation of different hydroxy. There is some sound commented. Hydroxy derivatives. So it can also happen modification on the head group of this lipid, for instance, non-enzymatic glycation or Maillard reaction with the formation of glycated derivative. So a few years ago back, we actually introduced a term which we call an epilipidom to sum up all this kind of different modifications, which we call a subset of natural lipidome formed by lipid modification, enzymatic or non-enzymatic ones required to regulate complex biological functions. So in principle, if you think about this is something pretty much established in the other omics field when we talk about DNA methylation. So this is also small chemical modifications regulating epigenomic events, protein post-translational modification forming all those proteoforms. So we can call this modification of lipids are the lipid PTMs composing this epilipidome to gain in regulatory capacity of biological systems. So the examples of epilipidome regulation are all around us. So we heard a lot about oxylipids and oxidized fatty acids, where there's a lot of established in this field, but complex oxidized lipids are also known to regulate many activities, for instance, especially in activation of innate immune responses, for instance, or formation of this lipid whiskers and activation of pattern recognition receptors. There are some data coming up about the role of oxidized lipids in different cell death events in the apoptosis, but also now it's a very popular field of research, the oxidized lipids in ferroptotic cell death. So these oxidized lipids are not secreted in a free form as eicosanoids, so they remain bound to the membrane and then facilitate the activities in this way. So what kind of modes of action can be attributed to them? For instance, low affinity interaction with proteins and membrane remodeling. So these oxidized lipids can remodel and alter membrane polarity, its electronegativity, and in this way also change the properties and activation of membrane-bound proteins. So what we know that if we form different kinds of modification, just simple hydroxylations, but also this truncation events, this more polar modified chain can actually pop out of the membrane and form what is called lipid whiskers. Those lipid whiskers can in turn be recognized by pattern recognition receptors. In this way, they can mark damaged senescence and dying cells. They can also be present on the lipoproteins, and there is a whole sequence of events which are activated by the recognition with the receptors, for instance, in macrophages. So, but it's not so clear so far all of them are pro or anti-inflammatory, and perhaps it's not either of those. It's very context dependent, depends on the cell type of site of generation and the type of the modifications. So we can say that they can play anti-inflammatory effects when they're recognized by the resident immune cells, for instance, for non-immune clearance in the silent waste disposal. And there is a really nice review paper from Valerie, so where you can read more about that. But they also, when accumulated in the higher levels, they can act as a pro-inflammatory molecules and participate in the development of inflammatory responses. So it's definitely a very interesting class of lipids, and we have to think about how we actually can get access to this information and describe this epilipidom in the native conditions. So the analytical strategy, and I will just describe you here steps which we undertake here for this kind of identification. So the analytical strategy includes several steps. So first, of course, as we already talked here about several times about their extraction and storage. So if we look for the oxidized lipids, but I guess for lipids in general, we should take care about that. So we always store our samples and lipid extracts afterwards, and the temperature is -80 or below, avoiding any freeze and throw-in cycles, and we use radical trapping antioxidants in all extraction solutions and the storage solutions as well. For instance, BHT is very useful. So after we extracted our oxidized lipids, we have to decide on our LC-MS workflow. And so the question here, should we do it with chromatography or without? And if with chromatography, which kind of chromatography we want to use? So these oxidized lipids in the natural conditions are very low abundant. So they will be masked by highly abundant unmodified ones. So separation by chromatography will help us a lot to improve sensitivity and specificity of our analysis. Moreover, these oxidized lipids represented by a really huge number of isomeric species. And if they are co-fragmented in our tandem mass spectrometry experiment, that limits our specificity of identification. So we strongly rely here on separation by reverse phase chromatography before mass spectrometry detection. And this is just an example here, the standard lipid PAPC oxidized for different time points. And you could see with the different points of oxidation, we start to accumulate in the earlier retention time, so eluting earlier from the reverse phase products corresponding to the oxidized species. This is pretty logical because if you remember what I mentioned in my first talk, so reverse phase separation based on the hydrophobicity, we can express it as a log P values for each lipid species. And for instance, unmodified PAPE has a log P around 11. So with introduction of different oxidized group, hydroxy groups here in different way, we decrease log P value. So those analytes will definitely look earlier from our reverse phase chromatography. So in terms of mass spectrometry methods which we can choose, there is, not so clear how to go. From one side, so they are low abundant. That would mean we want to go for the targeted solutions, like multiple reaction monitoring or parallel reaction monitoring. The plus in this way, it's specificity, because as I mentioned and other speakers talk, we have to have this predefined list of analytes, so are very specific here and sensitive. The data is easier to process because we do not need really to identify them as we know them before. But on the other hand side, since we deal with predefined list of modified lipids, how do we know that we didn't miss anything? Yeah, how do we know that this list is actually specific for our conditions, for our sample? So on the other hand side, so these lipids are very, very chemically diverse. And if we want to address this unknown sample specific epilipidom, we usually would go for discovery or untargeted solutions. So as we mentioned again today, so data dependent or data independent acquisition methods. The plus here that we really can address this large coverage of oxidized lipids, find the new one and previously undiscovered one, address really the sample specificity. But on the other hand side, these untargeted methods would be definitely have lower sensitivity, and we will have much more complex data processing because we have really actually to identify all of them. So what we came up here is a combination of analytical and bioinformatical solutions where we first identify a native lipidome in our particular matrix of interest, so blood plasma, tissue, or cell extracts. And afterwards, we use this native lipidome to perform in silico oxidation. So for that, we use our software tool, LPP Tiger, which allows us, one of the part algorithm behind that allows us to predict oxidized lipids, which will be specific for this particular lipids of a native lipidome. And Lipid Tiger is doing that not by simple enumeration of the oxygens, but behind this algorithm stands such reconstructed metabolic networks, which we build it up based on the literature data for 10 polyunsaturated fatty acids. So in different ways how they were detected to be oxidized. So LPP Tiger will build us the specific, for this sample, specific epilipidom, and we will use it in semi-targeted LC-Msms analysis using this predicted precursors as inclusion list for data-dependent acquisitions. So then we can use again LPP Tiger or manual identification to confirm our lipids, and that will allow us to have this identification of the sample-specific epilipidom composition. So it works like that. We take some lipids which we identified as unmodified, specific to be in the sample with their corresponding fatty acyl chains. We use it for predict oxidized lipids by LPP Tiger. We get a huge list, but we can use still in the semi-targeted approach I described you here. So we use it as inclusion list and data-dependent acquisition. So we look for oxidized phospholipids, oxidized cholesterol, ester, and STGs, and we can do it in the one method by using polarity switching because they are very nicely separated by mass spectrum or by reverse phase chromatography. And then we go for the identification. So I would like to focus here that we look for the different levels of confirmation of our lipids, so in the MS1 and the MS2, but also retention times. So to show you how exactly step by step we do that, so first we look on the MS level. So we need to identify our signal corresponding to the potentially oxidized lipid. So we have a defined mass to charge ratio, which we measured, and we know that this precursor was triggered by our inclusion list in the DDA into the Msms events. So we check isotopic distribution to make sure that there is no overlap with the isotopolog signals of the lipid with one double bond or less. And since we are using high resolution mass spectrometry, we can define elemental composition for our lipid with provided mass accuracy, for instance, 3 ppm usually. So using that combination of this MS scan, so we have exact mass, so mass to charge ratio from which we can predict elemental composition. But here we can only just propose that that lipid can be maybe oxidized phosphatidylcholine with this combination of the carbons and double bonds or double bond equivalents and the 2 more oxygens. So another check, if we go manually, is retention time mapping. So we would like to check if actually that does it make sense. So as I told you that in reverse phase, any oxidized lipid will elute before its unmodified precursor, and more oxygens we incorporate in the structure during oxidation, the earlier they will elute. So with our experience now, of course, we also know the elution orders, for instance, that hydroxylated isomers elute before epoxy derivatives or 2 hydroxy groups before isomer with hydroperoxide. So we also know that the positions of different isomers now also define their retention time. But simply speaking, here we look for our ion of interest, which we defined before, shown here in red in extracted ion chromatogram, and the unmodified counterpart of this lipids shown here in green. So we see, okay, it's definitely eluded before, that makes total sense, and we see different kind of isomeric species separated by our reverse phase chromatography. When all of that works, we can go for MSMS level of identification, and we start with a lipid class and fatty acyl chain-specific identifications. So we look for the fragment. So we think maybe this is the lipid which we have. So it's a PAPC lipid with a hydroperoxide group. But how do we know that? So first we look for this kind of ions which are specific for the phosphatidylcholine group, presence of this group in the negative ion mode, for instance, the loss of methyl formate, which is always happen for the formate adducts of PC lipids. So, okay, now we can confirm this lipid as PC lipid with maybe these compositions. And we also have a set of ions which confirm our acyl chains. So the one here at 255 confirms presence of fatty acid 16 to 0. The one at 335 confirms presence of 20 to 4 with 2 more oxygens, and we also have some lysyl fragments. So the next level is modification-specific identification. So we can look further and we see the specific behavior. So for instance, if we have a hydroperoxide, it's very typical to lose the water with the formation of corresponding cathode derivative. And the signal is usually much more higher because this hydroperoxide is very labile. So this loss of water happens very, very prominently. So we have this water loss ion much more intense than, for instance, hydroperoxide and here. We can also have some loss of hydroperoxide itself. So now we can confirm that with the presence of those ions that indeed we have PC lipid with 16,0, 20 to 4, and it carries a hydroperoxide group. So when we are lucky enough and we have a good intense MS2 spectra, we can even go further and look for the modification position specific identification fragments. So these fragments here shown in the dark red are actually modification site specific, and we can follow the reaction of this gas phase fragmentation reaction mechanism, for instance, of the hydroperoxide ion with the formation of corresponding fragments. So having them present here, we indeed can say, okay, now we have phosphatidylcholine with this fatty acyl combination with modification corresponding to the hydroperoxide and the position 15. So the whole process of the confirmation for those lipids goes according to this annotation checklist. So we check elemental composition of proposed precursor using high resolution mass spectrometry. We check always in the complex data set that it's a really monoisotopic mass of our lipid of interest, and we don't have any isotopic overlaps here. And then we go step by step for the MS2 level confirmation. So for the lipid class specific fragment, for lipid molecular species specific fragment, modification type, and modification position specific one. So I must say, of course, I selected a nice example for you. So sometimes we cannot reach all those 4 levels of identification on the MS2 level from the real data set. So we have to stop somewhere here, maybe, or here. So, but then we report it on the corresponding level. And finally, we also make the retention time mapping here. And I showed you just one example, but when we use and look at the complex data sets and we have many lipids identified, dozens of them, then of course we plot them using Kendrick mass defect by hydrogen usually, but sometimes you can also do by oxygen here, which really helps you to eliminate some false positive identification specifically for the isomeric lipids, for instance, epoxy versus hydroxy and so on, because they have very defined retention time behaviors. So another example here, just to show you something else, we have, for instance, isomeric lipids here. We have again our MS1 spectra, where we can propose our elemental composition and get maybe idea what it could be. So then we look for the MS2, and that goes always the same way. So we look for lipid class specific fragments, in this case, again, confirming the loss of methyl formate from corresponding to the PC head group. We have our molecular species specific fragment confirming the fatty acyl composition in this lipid. In this case, we have an ion of singly hydroxylated arachidonic acid, which loses water pretty good. So it's a good indication that we have a hydroxy group here, not an epoxy. And then we go for this lower molecular weight fragments, which allows us in most of the cases define the position of a modification. Since we have this relatively good separation on the reverse phase chromatography of these different isomers, we are able to get this specific clean MS2 spectra from different isomeric compounds. So one more example here. So the same kind of precursor, the same extracted ion chromatogram, but now the peak, which is eluding later on at around 13.9 minutes here. So we have again our precursor, elemental composition. We look for the lipid class specific fragments, again, loss of -60 units here, corresponding to methyl formate. Here we even see some head group specific anion here. Then we go for the acyl chains. In this case, we have the same 319 ion, but it doesn't lose water at all, which is usually will be characteristic for the epoxide presence for this lipid. And then we look for this specific fragments which might explain us the position of the modification site along the chain. So in this case, we have another isomeric species here, which corresponds to the modification with epoxy group, which is located in the position 5,6 on the arachidonic acid chain. So, and in this way, we can principally assign all those fragments for different signals which we see in our extracted ion chromatographs. So, finally, at the end of my talk, so I would like to note that, of course, it's very time-consuming, but nevertheless, we went through hundreds and hundreds of such MS2 spectra to make sure that we are very familiar with our identification patterns. But of course, our aim to do it in the truly omics approach, so using the high throughput solutions here. And for that purpose, we use our software LPP Tiger. And if you are interested to know how does it work, there will be a tutorial provided by Jishuni on day 4 of our school, where he will explain how we designed the first version of LPP Tiger and how does it actually works. So now it works for phospholipids only, and that actually covers this step in our checklist. So we do not have the position specific fragments, but we can go up to the modification type specific fragments here. So now I would like also to mention that we are working very hard on the updated version of LPP Tiger, which we hope to release this year, which goes further on and also includes this modification patterns for modification position specific fragment ions. It will also include this retention time mapping capabilities, and we will cover not only phospholipid, but we also collected a lot of data for oxidized cholesterol esters and oxidized triacylglycerides. So finally, another important thing to mention that we are closely working together with Laura Garaci, who is responsible for the development of LipoStar software. So you might have heard about this tool, which is very popular nowadays for the identification and high throughput identification of modified lipids. So we joined our forces here that the signals, precursors, and corresponding MS2 spectra, which were proposed by LipoStar as potentially oxidized lipids, are automatically transferred into LPP Tiger, which is identifying them with a high accuracy and brought back into the LipoStar interface so that users can get both unmodified and modified lipids together. Okay, and with that, this was all what I wanted to show you, and I guess I have some time for the questions.